Hello students, today we will see regarding the remaining part of your preformulation. Now we will see regarding the crystallinity. Crystallinity is nothing but the crystal habit, and with the internal structure of any drug particle have the different type of physical properties. So any drug particles which is solid in nature may exist in the crystalline form. So when your drug is exist in the crystalline form, it may have the crystal habit and different internal structure. So crystal habit is nothing but the morphological description of outer appearance of the crystal, as well as the internal structure is nothing but the molecular arrangement within the solid particle. The changes in the crystal habit may cause changes in the internal structure, or changes in the internal structure may cause changes in the crystal habit of your drug. Example is the conversion of solid salt to its free acid form to produce the both changes in the internal structure as well as the crystal habit of that drug. Now the different shapes of crystals. How your crystals may exist in the different forms. There are the different shapes of your crystals are appear: cubic, isometric, tetragonal, orthorhombic, hexagonal, trigonal, triclinic, as well as monoclinic. So there are the various shapes of crystals may exist depending on the process of synthesis, depending on the inherent nature of your drug particle. So these are the different shapes of crystals are there: cubic, tetragonal, clinic, orthorhombic. Now crystal form. When there is a crystal, depending on its internal structure as well as the morphological nature, so your crystal forms may have the two types. First one is the crystalline form, another one is the amorphous form. Now crystalline form means what? When your crystal is having three-dimensional array structure, as well as it is having the fixed internal structure, so that is called as the crystalline form. And this crystalline form is more stable but have the very less solubility in water, as well as the less dissolution rate, as well as it is having the one advantage that it is having the lesser tendency to change its one form to the another form. So it not undergoes the polymorphism, or it is very lesser undergo the polymorphism. But in case of amorphous form, when your drug particle having the random arrangement of the structure, then it is called as the amorphous form, where it is not having the fixed internal structure, as well as amorphous form of drug is having the less stability, but it is having one advantage that compared to the crystal form, amorphous form is having the good solubility in water as well as the good dissolution rate, and it is having the higher thermodynamic energy. So one disadvantage is there: more compared to your crystalline form, it can have tendency to revert or to change from one form to the another form, but it means it undergoes the polymorphism. Now what is meant by the polymorphism? When the ability of compound to get crystallized as more than one distinct crystalline species with the different internal lattice, it means change of one form to the another form, it is called as the polymorphism. The different crystalline forms are called as polymorphs. When it is existing into the different forms, then it is called as the polymorphs. The polymorphs differs from each other in different physical properties. One polymorph may have the different solubility, melting point, density, hardness, and compression of this another form of the same drug. It means if the one drug is getting changed into the other form, it means drug is same but its physical characteristics may be differ from one form to the another form. During the preformulation study, it is important to identify the polymorph that is stable at the room temperature or not. Then only it will be essential or important while manufacturing of that dosage form. Examples of such kind of polymorphs are the chloramphenicol, which is exist in the three forms like A, B, and C. So among the three form of that chloramphenicol, only the B form is very much stable and well as it is most preferable one. Regarding the another case is the riboflavin, which is having the 3 forms, or it is exist in the 3 forms, and its third form shows the 20 times more water solubility than the first form. So these are the examples of polymorphs. Now the polymorphs are of again 2 types. There are the enantiotropic as well as the monotropic. So what is meant by enantiotropic polymorph and monotropic polymorphs? The polymorph which can be changed from one form into the another form due to the change in the temperature or pressure is called as the enantiotropic polymorph. So example is the sulfur. It means the polymorphism occurs when your temperature is get changed or pressure is get changed. So that kind of drug or that kind of polymorph may be called as the enantiotropic polymorph. Another polymorph is those polymorphs which are unstable at all temperature as well as the pressure. They are called as the monotropic polymorphs. Example is crystalline stearate. Now these are the methods which we are utilizing for the studying your different crystals, their morphological form, their melting point, transition state. So to study your crystals, we can utilize some analytical tools. They are microscopy, hot-stage microscopy, thermal analysis, X-ray diffraction, IR, PMR, NMR, and scanning electron microscopy. So what is the microscopy? So microscopy is very well known and it is handled by everyone. Okay, microscope is used to study the crystal morphology and different between the polymorphic forms. So here we can use the simple microscope to check or to find out the morphological as well as upper characterization of your crystal, as well as we can check the difference between one polymorph to the another polymorph in case of its morphological character. Now hot stage microscopy is a little bit modern type of microscopy. So the microscope with the fixed hot stage is used to study your crystal. Generally hot stage microscopy is utilized to study the polymorphism, melting point, transition temperature. In this technique there is one disadvantage, that as we are increasing here temperature for the studying purpose, so during the study your drug may get burned or it may get degraded because of the increase in the temperature. This is the picture of the hot stage microscopy. Now thermal analysis. There are the various technical tools, analytical tools we are using to study your drug. So by utilizing the thermal or the heat, we can study your crystals. So first analytical tool is the differential thermal analysis. It is called as shortly DTA. This technology used to study your crystals. In this DTA study, temperature difference between the sample and reference material is recorded and it is plotted against temperature. So by utilizing this principle, we can study the various crystal forms as well as amorphous forms. Generally, the standard or the reference material used in this study is the alumina as well as the Kesselgar. Next technology is the differential scanning calorimetry. Differential scanning calorimetry measures the difference in energy inputs into the sample as well as into the reference material. So it is measured as the function of the temperature. The samples we can study by using DSC are powders, some fibers, single crystals as well as polymer films can be studied. Now the application of DTA as well as DSC. Generally DTA as well as DSC can be utilized to determine the number of polymorphs of your crystal as well as to determine its ratio, as well as it is used to determine the heat of salvation It is also used to determine the purity of your sample. It is also used to determine the thermal degradation of drug as well as the excipients, as well as it can be utilized to determine the glass transition temperature of a polymer. After that, we can have the another tool, that is the thermogravimetric analysis. It is shortly called as the TGA. TGA works on the principle that it measures the changes in sample weight as a function of time as well as temperature. So at the time gets changes, at the temperature gets changes. So what is the difference of weight is occurred in the sample. Generally, TGA, it is used for the dissolution and decomposition process, as well as the data recorded by this study can be used for the explanation of thermal process. Another tool is X-ray diffraction tool. So this tool is used to find out the structure of your crystal, whether it is crystalline or amorphous. In this technique, we used to impart a beam of non-homogeneous X-ray light through the crystal. The X-ray beam is diffracted due to the three-dimensional crystal, and it is recorded by means of photographic plate. The random orientation of crystal lattice scatters rays into the reproducible pattern of the high intensities. The diffraction pattern is characteristics for a specific crystalline lattice for a given compound. The X-ray provides the most complete information about the solid state of the crystal. An amorphous form of the crystal does not produce such kind of measurable pattern. This is the instrument generally we are using for the studying the crystallinity. Now, the next part is the degradation pathways.